To open the first AOS special session, may we call on Professor Yeul Hong Kim, our AOS President, and the Director of the Korea University Cancer Institute and Korean Precision Medicine Enterprise. He is a professor of medical oncology at the Korea University College of Medicine, and is a pioneer in the area of liquid biopsy, using circulating tumor DNA, isolated from cancer patients' blood. Please welcome Professor Yeul Hong Kim.
Good afternoon, dear colleagues. It is my great honor and privilege to chair the first AOS special session. My name is Yeul Hong Kim. I'm a professor of medical oncology in Korea University in Seoul, Korea. And also I'm a president of AOS. In this session, the title of this session is Precision Oncology in Asia. The precision medicine is most updated art of our clinical science. So in this session, we have three very famous, prestigious speakers from Italy, Japan, and China. And I would like to introduce briefly those three speakers. The first speaker, who is Professor Alberto Bardelli, is from full professor, Department of Oncology in University of Torino in Italy. And his work is aimed at developing precision medicines for patients with cancer. Professor Bardelli is a world-renowned expert in liquid biopsy research area. And now these liquid biopsies are presently used to monitor patients' response during treatment. And the second speaker is Professor Katsuya Tsuchihara. Dr. Tsuchihara is a chief of Division of Translational Research, Exploratory Oncology Research, and Clinical Trial Center in National Cancer Center, Japan. And Dr. Tsuchihara's main researches are focused on high-resolution genomic-wide analysis of cancer genomics to define mutational landscape of the cancer genomes using NGS. And the third speaker is Dr. Jin Li. And Dr. Jin Li is the director of Department of Oncology in Tongji University and Shanghai East Hospital. He is also president of Chinese Society of Clinical Oncology, and he is also chairman of Federation of Asian Clinical Oncology. His clinical specialties are comprehensive treatment of GI cancer and cancer immunotherapy. He's conducting more than 100 clinical trials for the treatment of various solid tumors, and including more than 10 global clinical trials. So I would like to call the first speaker first. Dr. Bardelli, please.
Hello, good morning. It is a pleasure to be with you today. Unfortunately, due to the current situation in the digital format, but my name is Alberto Bardelli. I'm a full professor at the Department of Oncology, University of Torino Medical School. And I'm also director of molecular oncology at the IRCC Institute in Candiolo, Torino, Italy. Today, I will be discussing with you some exciting data in the space of colorectal cancer metastasis, clinical trials, response to therapy, and DNA repair. So it is a great pleasure today to discuss with you DNA repair alteration and immune surveillance in colorectal cancer. These are my disclosures, one of which is relevant and is related to Neo4. I will mention this again when the time comes during the talk. My laboratory has been working for many years on colorectal cancer, which is a tumor type that has been extensively studied and that is described here. Starts from a single cell that acquires alteration in a specific pathway, the APC beta-catenin pathway, and then evolves through several histopathological steps that are described here, which are associated with genetic alterations. The final and most dismaying outcome is the development of metastases, which often occur into the liver, but also in other organs such as a lung brain and the peritoneum. So today I will discuss with you the role of somatic alteration in this tumor type, and there are two types of alterations that contribute. Those that are associated to tumorigenesis, they have a key role in therapeutic resistance, and they are determinants of intrapatient heterogeneity. However, often the very same mutations are also involved in being targetable, so that some of them are actionable target. Importantly, they tend to be present in somatically, so they are only present in the neoplastic cells, of course, with some exceptions. So they are not present in the normal cells of the patient, of the individual. And some of different types of DNA repair alterations lead to different type of mutations. This is very important. It's become more relevant recently. Importantly, for what I will discuss today, somatic mutations can be recognized by the immune system as non-self, as neopeptides, as we will discuss in a second, and this will trigger cancer immune surveillance. So today I will focus on these points that are highlighted here in red, and I will discuss specifically how some of these mutations can be exploited for therapeutic purposes when they are associated to DNA repair deficiencies. We all know that tumors tend to be very heterogeneous. There is intra-patient heterogeneity, there is inter-patient heterogeneity, and it's very relevant to what I will discuss today. In CRC, this is no exception. You can see here that the facts about CRC, it affects both women and men. It's the second bigger killer, and you know, a number of patients that develop the tumor over their lifetime will also develop metastasis. In fact, about 50% of them will develop metastasis. What can we do when surgical removal is not an option, or when surgical removal needs chemotherapy or other form of therapy? You can see that over the years there's been progress. Most recently, the addition of targeted agents such as EGFR blockade, or more recently even the addition of trastuzumab and lapatinib, to which we contributed. This is a subset of colorectal cancer that benefit from this type of therapy. There is also the possibility to use anti-angiogenic therapy. But essentially, the truth is that these targeted agents have done limitedly to change the outcome of the disease. So a few years ago, we became fascinated by the features of a subset of colorectal cancers. So those tumors that display mismatch repair deficiency. As you can see here in stage 1 to 3, these are about 15 to 20% of the cases. So a relatively significant number. However, if we go to the metastatic stage, to stage number 4, this drops down to 3 to 5%. And my group has been historically interested in understanding why this happens. And it's clearly the mismatch repair status. This type of DNA repair deficiency is associated with a better prognosis in colon cancer, possibly in other tumor types, but most importantly, this subset of tumors are very responsive to immunotherapy, immunocheckpoint blockade, as it is highlighted here. You can see that the best responder in general to immunotherapy are those that have either mismatch repair deficiency in one allele or biallelic mismatch repair deficiency. This is a very rare syndrome that is very fascinating. Also mutations in poly or other DNA repair pathway genes. So Giovanni and Monica in my group a number of years ago wanted to understand better why mismatch repair deficient colorectal cancer and other tumor types have this specific feature, namely that they had better prognosis and responded to immunotherapy. And so we focus on the genes that are involved in this pathway. I mentioned earlier there are several. And so the DNA repair pathway that involves mismatch repair has been conserved historically very much during evolution. You can see here the key players in this pathway. And they essentially are mutase and mutel, the same complexes that are present in bacteria. So there's been a conservation of these genes for millions and millions of years. And because the reason is that they are clearly central in replication of DNA in the cell and ensuring that replication occurs properly. In colon cancer and other tumor types, these genes are altered, as I mentioned earlier, and the genes that is most frequently altered is MLH1. And so we decided to replicate what happens in patients by inactivating MLH1, which is often epigenetically silencing in human tumors. And therefore, we decided to delete the genes so that we abrogated the expression. You can see here we took mouse colorectal cancer cells, we inactivated MLH1, and did a number of controls, including the so-called MSI status check. And you can see here that microsatellite in these tumors that have been modified to lack MLH1 shifted to the left. It's a very similar test to the one that is performed in patients, suggesting that we had, in fact, replicated the phenotype and the genotype that was observed in patients. Fascinatingly, if we take these cells and we either grow them in vitro or inject them in immunocompromised or immunodeficient animals, they grow pretty much like the parental cells. So mismatch repair deficient and mismatch repair proficient cells have the same properties in animals that have no immune system. However, when we injected these cells into immunocompetent animals, you can see here that the cells were either did not grow or grew very poorly. And you can see these very long survival curves, which suggest that indeed this phenotype was very remarkable. And it's very similar to what, in a way, it is observed in the early stage of mismatch repair deficiency in humans. That is, there is some sort of immune control. We wanted to test this even in a more selective model. So we selected pancreatic ductal adenocarcinoma, and we did the same experiment. We deleted MLH1, just like it happens in humans, and then we injected them orthotopically in immunocompromised animals or immunocompetent mice. And as you can see here, these cells were largely unable to form tumors, again suggesting there was a profound effect of inactivation of mismatch repair. So the next we ask is, what are the functional bases of this phenotype? Why are these cells that have no capability to do mismatch repair so effectively rejected or respond so effectively to immunotherapy? And what we learn is that a key player in this program, in this phenotype, are T cells, CD8 particularly, T cells. If we suppress T cells in immunocompetent animals, the cells are able to grow. We recently repeated these experiments and confirmed that CD8 cells play a role, but also found a role for other immune cells, and these data are still maturing, but are very exciting. Because we could not effectively form tumors with mismatch repair colorectal cancer cells in immunocompetent animals, we decided to first inject them in immunocompromised animals, form the tumor, transfer the tumor to immunocompetent, and if you do that, then the mouse is unable to control completely the tumor. As you can see here, there is still a delay, very prominent in fact, if you inject the cells in immunocompetent animals, but it does form a tumor. Why this is important? Well, this is important because then you can study immunotherapy in this setting. As you can see here, if we inject the same cells that I just described, first in immunocompromised animal and then transfer them in immunocompetent animal, and then we give the immunocompetent animal inhibitors of checkpoint, like anti-PD-1 or anti-CTLA4, we control the tumor, and in fact, we reduce massively the growth of these tumors, and sometimes, in fact, quite often, we cure the mice, which was remarkable. So these were very large tumors, over 500 in size. As you can see here, the tumor size drops, and at the end, there is no tumor left in the mouse. The next step that we want to address is, why are mismatch repair deficient cells rejected, and what can we learn from this phenotype, from this process? Is immunoediting involved? And so what we did is to remove MLH1 and then wait 130 or almost a year, and then looked at the profile of mutations in these tumors. As you can see here, over time, there is an increase in the number of alterations, and some of these alterations turn into neoantigen. So what happens here is we have neoantigen evolution, the development of new neoantigens, of neoantigens during time. And we think that this is very important because the immune system is likely recognizing them and sort of attacking the tumor. You can see that we can speed up the process in two ways. One is by waiting long enough and then checking for the phenotype. You can see that over time, the phenotype it becomes more prominent until the cells also not grow in immunocompetent animals. But the other possibility is to clone the cells, so generate clonal neoantigens. In this case also, you can see that there is a reduction in growth. And this bottleneck is very important to achieve that effect. The next thing that we ask, which is currently unpublished, is rejection dependent on antigen presentation? And as you can see here, the way we address this question is by removing genetically HLA presentation. And we did this by inactivating the major histocompatibility complex number 1. In order to do that, we deleted beta 2 microglobulin that is part of the complex. And without beta 2 microglobulin, the major histocompatibility complex cannot be presented on the surface of the cells. And so what Giovanni did and Monica together did is deleting beta 2M. You can see that on the surface of cells that have deletion, there is no MHC class 1, suggesting that these cells are unable to present the antigen. And excitingly, if we delete mismatch repair deficient cell beta 2M, so the ability to present the tumor, to present the new antigens, the tumor grows back, suggesting that again antigen presentation is critical for the phenotype that we observe. And excitingly also, and we still don't understand completely this, I recommend that you don't post this slide because it's important, it's unpublished, but you still have, as you can see here, even in the absence of antigen presentation, immunoresponse. And this is very alike to what happens in patients. In patients that have mismatch repair deficiency, colorectal cancer, and also have beta 2M inactivation, so no ability to present the antigens on the surface, they respond to immunotherapy. So we think this model sort of recapitulates what happens in patients. We're still studying this. We think it's very relevant clinically, and we will stay tuned because we may have more data soon. So the next step that we are asking, and this is still work in progress, is possibly a very high risk, high gain approach. So we thought that perhaps we can exploit therapeutically inactivation of mismatch repair. And in order to do this, and I have a conflict here, we decided to pharmacologically block the pathway for therapeutic purposes, something that you would not consider to do, that you would even imagine that would be too risky to perform. And of course, this could be very risky and could lead to nothing, but nobody had developed mismatch repair protein inhibitors before, which is something that we are actively doing with the idea that we will generate errors in tumor cells, specifically indels and frame shift that are key to generate the phenotype that I described. I didn't have time to get into the details, but mismatch repair deficiency will generate indels and frame shift that will generate new peptides that are very long. And so this is exciting. And in order to do this, we started up a company that is called Neo4, and you can see here the concept that is behind it. We want to block mismatch repair inhibition in order to increase mutagenicity, in order to increase mutational burden, and enhance the effect of immunotherapy. And of course, this is a long process. Nobody had done inhibitors of this pathway before. And so it took us a couple of years to figure out how to do it. We have, I'm pleased to report that we have now several compound leads in this space, and we're testing whether they have a phenotype. And we are very excited by the process because it suggests potentially a new way to inhibit tumor growth by targeting the cancer cells and then helping in that way them to be recognized by the immune system and fostering immune surveillance. In the second part, apologies again, in the second part of my presentation, I will describe something that is very recent, very exciting, still in the space of colorectal cancer, of course, and other tumor types and neoantigens. And so one thing that we ask is having learned from mismatch repair deficient tumors, we asked whether there was another way that we could use to boost immunosurveillance. And so let me switch a little bit gear, and you may think, why are we seeing oranges here? The reason you're seeing oranges is because we became excited by some recent data in the space of vitamin C and its role on as an anti-cancer therapy. And in fact, the recent data are the one that I will describe to you, but they relate back to a story that has been going on for decades, and you may know. But let me summarize briefly to you, if you're not very familiar, what is vitamin C. Vitamin C is an essential vitamin. It's a vitamin, so of course it's it's essential. It's water soluble. We assume vitamin C with the diet. Leukocytes accumulate very high concentration of vitamin C. Is a cofactor of type deoxygenase and histone demethylases. But also very importantly, it is very well known that chronic vitamin C deficiency is associated with ineffective immunity. And of course, everybody knows that without significant level of vitamin C, humans that cannot produce it will go and face scurvy, which is a disease that has almost disappeared, but it's very important to remember that scurvy patients also have immunocompromised functions. So I told you that we became interested in vitamin C because of some recent data. You can see the data here. The group of Luke Cantley and others have reported that vitamin C is able to block the RAS-BRAF signaling pathway in colon cancer cells. And because we were studying colon cancer cells, we wondered whether we could understand better the impact of vitamin C. Of course, this paper is related to the cell autonomous impact of vitamin C. And in order to explain what we found, we need to go back a number of years to the wonderful data of Linus Pauling. You may know, I'm sure you know about Linus. He was a chemist. He won the Nobel Prize twice, once for chemistry and one for literature and peace. And his data on vitamin C have been controversial. You may remember that in the 70s, Cameron and Pauling reported some very exciting data on delivering vitamin C intravenously to terminal cancer patients and seeing some remarkable efficacy. And you know, these data, of course, made news back then in the 70s, as I said, but unfortunately, a follow-up study done by the Mayo Clinic saw no benefit. However, as you can see here, there was a profound difference between vitamin C that was delivered orally by the Mayo Clinic and vitamin C that was delivered IV by Cameron. And this is very important, and we will discuss in a second why this is. Later, there were many studies on vitamin C, even more recently, and you know, these studies are inconclusive, controversial. Most of them tend to be negative, but sometimes they reported some results. But importantly, what we think that is very crucial, and what I will remark again in a second, is that the plasma level of vitamin C are critical to see the phenotype that I will describe in a second. You can see here that if you deliver a vitamin C orally, you can reach micromolar concentration. If you deliver vitamin C intravenously, you reach millimolar concentration. You can see here what happens with the dosing of vitamin C intravenously or orally in the plasma of animals or patients. And this has been studied both in mice and in humans. In concluding this brief introduction on vitamin C, I'd like simply to say that the National Cancer Institute currently has not approved, does not recommend the use of vitamin C. However, has also recognized that some data have been obtained, and so this remains very controversial. However, this is not a standard therapy, that's for sure, for cancer. Let me tell you why we became so interested in vitamin C. You can see the data right here. So when we started to grow tumor cells, mouse tumor cells in immunocompromised and immunocompetent animals, as I described in the first part of my talk, and then deliver vitamin C intravenously, we did see a major difference in the number of tumor types. These are breast cancers, these are colon cancer, and these are melanoma. We also studied PDAC, which is a pancreatic ductal adenocarcinoma. And so you can see that in most of the models, not in all, but in most of the models, the single administration of vitamin C delayed tumor growth. Now, vitamin C is obviously not a drug, and so I was personally surprised that a vitamin had this prominent effect. And so we decided to follow up this further. And the first thing that we did was to verify whether what was the dosage that was needed to achieve this level. And you can see here that only dosing vitamin C intravenously was able to achieve the inhibition that we were observing before, comparing immunocompromised and immunocompetent animals. And so the next step was then to try to understand why vitamin C was playing this role in delaying tumor growth. And what we observe is that vitamin C enhances the expression of interferon gamma. in CD4 and CD8 T lymphocytes, suggesting that there was an impact not only or not predominantly, in fact, in the tumor cells, but in the immune cells of the mouse. And similar experiments to the one that I described before were performed by Alessandro, the first author of the study, and he tried to suppress T lymphocytes and saw that they were involved in the vitamin C response, mediated response. And so we suggest that this efficacy is remarkable and is largely to the immune system. We still are pinpointing which cells of the immune system are involved in the response. Even more remarkably is what we observed when we gave vitamin C, again, intravenously together with immunotherapy. And you can see here that we observe a profound delay, sometimes tumor regression even, when the two were combined. And you can see that in some cases, we had mice that were tumor-free after a number of weeks, suggesting that the effect of vitamin C was remarkable. We also observed that when vitamin C was delivered and immunocheckpoint blockade was concomitantly delivered, there was increased infiltration in the tumor, suggesting that this was really activating the immune system and allowing the immune system to counteract the tumor by infiltrating the tumor. And so the next and final data that I want to show you today is what happens when we combine the two stories. So we decided to then look at mismatch repair deficient tumors in the presence of vitamin C. Perhaps we thought that the two could complement each other. And first of all, we did see that vitamin C again had no impact on mismatch repair deficient tumors when they were growing in immunocompromised animals. However, the addition of vitamin C was remarkably effective. Of course, again, this is just a vitamin, okay? So we're delaying tumor growth by giving the vitamin, not giving by a therapy that may be toxic. And you can see here. However, even more remarkable and potentially really astonishing was the effect that we observed when we combine mismatch repair deficient tumors in immunocompetent animals with vitamin C with immunotherapy. And here we did see some, we did cure many mice, which was remarkable and suggested to us that perhaps this could be exploited in the clinic. And the final piece of data is that the mice that were treated with vitamin C and immunotherapy and that were tumor-free, when rechallenged with the tumor, were vaccinated. Essentially, they rejected the tumor off the bat, suggesting that the immune system of these mice had been reprimed and was ready to recognize the tumor. And similar experiences were observed when we did adoptive transfer of the tumor cells. So I'll conclude by saying that I presented to you some data on colorectal cancer and other tumor types and DNA repair deficiency, specifically mismatch repair deficiency. I also mentioned some exciting results that we recently obtained in the space of vitamin C modulation of immune response, either as a cell autonomous, but most importantly for us, as a immunodependent phenotype. And we're still studying these. Of course, these can be exploited in the clinic. So we are designing, in fact, we have designed a clinical trial whereby we will combine DNA repair proficient and deficient patients, and they will receive vitamin C together with immunocheckpoint blockade. And with that, I'd like to thank the people that were involved in this study. This is my group. You can recognize some of the names that I mentioned earlier. Giovanni Germano, Monica Lamba were involved in the mismatch repair deficient study. Sabrina, Arena, and Nicole are studying DNA repair deficiency in colon cancer in my group. There is a large team of bioinformaticians in my group that you have seen. They will provide this very exciting new antigen-based data. And all the clinical collaborators that are listed here are very much engaged in what we do. And if you want to know more, you can log in and check us on Twitter or on the web. Thank you very much for your attention.
Hello, everyone. I am Katsuya Tsuchihara of the National Cancer Center, Japan. I appreciate the organizers for giving me this opportunity. So today, I would like to introduce the research and development platforms for precision oncology in Japan. First, I will talk about genomic testing systems, both companion diagnosis and comprehensive genome profiling test for daily clinical use. Then, large-scale academia-led research projects in this field. Genomic testing has been actively incorporated into cancer care in Japan in the past decade. Here, I show the rate of EGFR testing in patients with advanced or recurrent non-small cell lung cancer. In Japan, more than 90% of patients have undergone EGFR testing. This high testing rate is due to the high rate of EGFR mutation positive patients. In addition, companion diagnostic kits have been approved by the regulatory authorities for use at any hospital in Japan. And these tests are reimbursed by public health insurance. So the financial burden on patients is low. Since the approval of the PCR-based KRAS kit for colorectal cancer in 2012, a number of companion diagnostic kits have been made available for practical use. And in 2018, an NGS-based diagnostic system capable of simultaneously detecting multiple biomarkers for lung cancer was approved. As well, comprehensive genomic profiling systems for solid tumors were approved in the same year. Currently, 2 NGS panels using tumor tissue samples are approved and reimbursed in Japan. Both tests cost approximately $5,000 US, and the patient pays 30% of the cost. While Foundation 1 is also approved in the US, the NCC OncoPanel was developed at the National Cancer Center Japan and was approved for marketing by Sysmex, a Japanese medical device manufacturer. In addition, a liquid biopsy system has also been submitted for approval this year, and we will be able to use it soon. At the same time as approving the testing system, accredited hospitals across the country that can perform comprehensive genomic profiling tests. Because profiling tests are technically very difficult, we have established uniform standards across the country to ensure that the same level of testing is carried out. Lab-developed tests are not officially allowed, and we are recommended to use regulatory approved systems. We have also standardized the informed consent system and the genetic counseling system. These are all done from the perspective of patient protection, and these are the foundation for making genomic testing an affordable care and best practice. However, because the number of medical institutions that have such specialized personnel and equipment is limited, we have formed a network of 3 layers of hospitals. Because of the companion diagnostics have been widely used for standard of care in Japan, the genomic profiling test is targeted at patients with solid tumors that have progressed on standard chemotherapy, rare tumors, or tumors of unknown primary. We expect to test about 20,000 patients maximum in a year, and we will provide specialized annotation for all of these cases. We set up 12 core hospitals and 33 hub hospitals so far, where individual molecular tumor boards can be conducted. And 161 liaison hospitals will collaborate to recruit patients with core and hub hospitals. Ministry of Health, Labor and Welfare also established a central data center to consolidate the data obtained in the hospitals. We call this the CCAT, and the National Cancer Center is running it. The CCAT has a 4-part mission. First, to accumulate clinical and genomic information and provide standardized annotation reports to support molecular tumor boards. Second, to promote data sharing among accredited hospitals to help treatment decision. Third, to establish data utilization as a foundation for clinical and translational research. And fourth, to development human resources. A portal system to utilize the CCAT data is under construction. From September this year, it will be possible to search for data from core hospitals and assist the molecular tumor boards in making treatment decisions by searching for similar cases. From next year, the system will be available to academia and companies for research purposes. Ahead of these public infrastructures, a genomic screening network for clinical trials by academia has been in operation since 2015, a consortium called Scrum Japan. It is a collaboration among more than 270 academic institutions led by the National Cancer Center and 19 pharmaceutical companies. To date, we have performed targeted sequencing of more than 15,000 solid tumors and recruited patients into 56 clinical trials. As a result, 7 therapeutics and 7 diagnostics have already been approved in Japan. Scrum Japan has also been actively introducing liquid biopsy. Liquid biopsy has many advantages because it allows us to evaluate the patient's situation during treatment in real time. In fact, the number of patients participating in clinical trials has more than doubled since the introduction of liquid biopsy. Now, we are focusing in visible cancer as well as visible tumors. Beginning in 2020, we also started a large clinical trial in surgical cases, which we call Circulate Japan. In part A, we evaluate the residual cancer cells by liquid biopsy after surgery for colorectal cancer that has undergone a curative resection. Multiple clinical trials will be conducted based on these data. This is a scheme of the observational study called the Galaxy study, which enrolls 2,500 patients and periodically follows up the residual tumor by customized liquid biopsy panel. The results will be shared with global research groups to create a database of tens of thousands of cases. Recent studies suggested that liquid biopsy evaluation of residual tumors can predict the recurrence of colorectal cancer with a high probability. In the VEGA trial, we will randomize patients with ctDNA negative tumors and investigate whether postoperative chemotherapy can be omitted. Besides, in the ALTAIR study, we will follow ctDNA positive cases after adjuvant therapy, and we will see if additional treatment is needed. Finally, the Ministry of Health, Labor and Welfare is planning to conduct whole genome sequencing and multi-omics analysis of 100,000 cancer cases in the next 5 years. On the other hand, Scrum Japan is moving to its 4th term project in 2021. We will conduct wider multi-omics analysis and more rapid turnaround of data sharing with multiple stakeholders. We hope that this foundation of precision oncology will contribute to cancer treatment development in Asia. Thank you very much.
Dear Chairman, dear colleagues, my name is Jin Li from Tongji University, Shanghai East Hospital. Many thanks to the organizer of this meeting to provide me the opportunity to give a talk here. The topic of my speech is the targeted therapy for MCRC. The colorectal cancer represents a significant health issue as it is the most common GI tract cancer worldwide. It is the third most common cancer diagnosis and the second leading cause of cancer death. There are 7 targeted therapies available right now. The large molecule monoclonal antibody, bevacizumab, cetuximab, panitumumab, and the ramucirumab. A small molecule inhibitor, including regorafenib and fruquintinib, is approved in mainland China. The very latest development is the anti-PD-1 antibodies in microsatellite instability high patients. Let's have a brief review for standard targeted therapy for MCRC. The first targeted agent for CRC approved by FDA was cetuximab in 2004. Followed by bevacizumab in the same year. From that time on, emerging targeted drugs for MCRC have been successfully brought to market to save lives of MCRC patients. With more on the way, numerous agents have been developed and brought into preclinical and clinical trials. Let's look at a trial in China, the FRESCO study. We analyzed the fruquintinib compared with placebo in the third line to treat the MCRC patients. The results showed the fruquintinib can prolong the overall survival as well as the PFS as the third line treatment when they refractory to at least the second line chemotherapy. Immunotherapy is another achievements in MCRC. Although the checkpoint inhibitor is not very active in the colorectal cancer, but for the patients with dMMR, the pembrolizumab, nivolumab can provide a higher response rate, longer OS, and the PFS. So that in the future, not only the checkpoint inhibitor could be used in the MCRC, but also can combine with ipilimumab to treat the cancer patients. The untracked biomarker is another very important issue for MCRC patients because the larotrectinib was approved by FDA for TRK fusion. In those patients, they got a response rate of 62%. Including 3 CRC patients all showed the shrinkage of tumor. The treatment options for MCRC continue to emerge. However, there remains a number of challenges to overcome. The communicated signaling pathways and the network crosstalk involved in tumorigenesis must be more effectively targeted. There's also the dynamic tumor microenvironment, the genetic instabilities, and the host immune responses to be better understood. Let's look at the RAS mutation. As many as 2,953 cases in Mayo Clinic and MD Anderson cancer, the analysis of the patients showed that the RAS mutation is a poor prognosis factor for MCRC. And also with the RAS mutated population, the patient seems cannot benefit from EGFR monoclonal antibody. I just list those 5 biggest phase 3 trial: OPUS study, Crystal, Fire3, CALGB 80405, and Taylor study. All of that, the studies showed the patients cannot benefit from RAS mutated population from the EGFR and monoclonal antibody. The BRAF mutation is another issue for MCRC. It is more refractory to chemotherapy, also refractory to combination with monoclonal antibody. The BRAF signaling is known to be implicated as an oncogenic driver in approximately 8 to 10% of CRC. So we have to resolve that problem. Another characteristics of HER2 amplification is also an issue for advanced colorectal cancer. The relationship between HER2 amplification and the progression-free survival is analyzed in some sets of clinical studies. The survival analysis showed HER2 amplification was associated with shorter median PFS on anti-EGFR antibody therapy compared with the HER2 non-amplified population. So to overcome these obstacles, many efforts have been done to provide patients a chance to survive longer and better. One trial is called the CodeBreak 100. In this study, AMG510 was tested as the phase 1 3+3 the dose escalation. In that trial, the response rate was showed very positive, and all the patients can tolerate to as high as 960 milligram of the dose. The PFS seems similar from the 960 milligram compared with lower dose. So in the future, if the patients cannot tolerate to higher dose of AMG 510, we can give them a reduction of dosage to a lower dose. Also can provide similar overall survival and progress-free survival. The other trial is the Beacon CRC study. In this trial, they combined the EGFR inhibitor, the BRAF inhibitor, and the MEK inhibitor. In this trial, the triplet combination of these three target agents showed a very good response, as high as 26% in the triplet combination, and 20% in doublet combination, compared with only 2% in control arm. So in the future, the combination of different target agents can be used as a treatment of BRAF mutated population. We can make what we achieved another achievements is the treatment strategy for advanced colorectal cancer with HER2 positive, the HER2 amplification. There are two trials, Heracles and MyPathway trial. Both of that trial was combined with trastuzumab with lapatinib or the pertuzumab. So both of trials showed a very good response rate, as high as 30% and 38.2%. So in the future, for the MCRC patients, if the HER2 amplification was found, we could treat them with the combination of trastuzumab with lapatinib or pertuzumab. Another target therapy for HER2 amplified patients is T-DXd. T-DXd is a novel antibody drug conjugate drug. This compound treated with patients with HER2 positive patients showed the confirmed overall response rate as high as 45%. So in the future, they will development of that drug for further phase 3 trial. I can predict that T-DXd could provide patients better survival time. This is my last slide. It depicts generalized treatment algorithm that is used in the management of patients with MCRC, further development of therapies aimed at membrane receptors, intracellular signaling molecules, and other protein kinase targets is ongoing. All of those potential targets demonstrate the complexity of cancer. So that means the MCRC is still a very hard nut for us to crack. But some promising breakthroughs have been made researching the role of different mutations profile in MCRC patients. As we move forward, further progress in identifying new targeted therapies with associated predictive biomarkers will be achieved definitely in the future. So thank you very much for your attention.
Good evening, ladies and gentlemen. This is again Dr. Yeul Kim. We have our speakers in here. Unfortunately, Dr. Li could not join here until now, but we have Dr. Bardelli and Dr. Tsuchihara. Nice for your, we are very thankful for your nice lectures. And we have a couple of questions from the audience. First of all, for Dr. Bardelli, there is a question about, maybe the audience is very interesting about your high-dose vitamin C plus immune checkpoint inhibitor application for improving immunoinfiltration of T cells. So maybe he asked about, do you have a plan for clinical trial, or is it already going on?
Yes, good evening, everyone. In Italy, it's a morning here, but it's a pleasure to join you. And thanks for the very interesting question and for the introduction. So, yes, indeed, our data in preclinical models of colorectal cancer and other tumor types suggest that, especially in mismatch repair deficient cancer, will benefit together with immunocheckpoint by the addition of intravenous high dosage vitamin C. Vitamin C has had a very long history in the treatment of cancer, sometimes controversial. It dates back to the time of Linus Pauling that won a Nobel Prize, as you recall. In fact, he won 2 for chemistry. So getting to your questions, yes, we have made very rapidly plans for a clinical trial. The trial has been designed. The difficulties has been with the formulation of vitamin C and how to dose these in the patients. But I'm pleased to say that the trial is called Alfeo and has moved forward. And so, yes, we will test these hypotheses in both MSS and MSI patients in combination with immunocheckpoint blockade. Yep. Thank you.
Certainly, the exhaustion of T cell is one mechanism of failure of immunologic drug in GI cancer. And also, there is another barrier for using immuno-oncology drug, because there is a stroma to prevent the infiltration of T cells. So do you have any plan for overcome those hurdles as well?
Yes, it's an interesting question. I believe that the stromal impact is particularly relevant in diseases like PDACs, like carcinoma of the pancreas, for example. It is also has implication for colorectal tumors, but interestingly, if you have a tumor that has very high tumor burden, mutational burden, I mean, and with a lot of neoantigens predicted, there is infiltration. So we think that in principle, this tumor type may be amenable for lymphocyte infiltration. Although I realize that until we formally test this hypothesis, we will not know.
Thank you very much. And Dr. Tsuchihara, very interesting presentation for your Japanese approach for precision medicine platform. And there is a question about utilizing circulating tumor DNA in your GI project. So could you explain why do you change or adopt circulating tumor DNA analysis in your platform?
Okay, so thank you very much. Of course, so we are most interested in monitoring the residual tumors after the surgery, or very first step of the recurrence of the colorectal cancers in our circulate the project. So, so in such case, we cannot detect these tumors in a radiological diagnosis. It means we cannot do any biopsies, tissue biopsies for these targets. So actually, the ctDNA monitoring is only one way to monitor such a small amount of the tumors. So that's why we are choosing the ctDNA systems. Of course, there is a very nice systems technologies to detect such a very high sensitivity the tumors.
Yeah. And since Dr. Bardelli is worldwide expert in the circulating tumor DNA detection, especially for colorectal cancer, we would like to share our information for this approach. Because in my experience, when we check the circulating tumor DNA content in colorectal cancer patients who had been treated with neoadjuvant chemotherapy or chemoradiation, the release of tumor DNA in the blood is very low concentration, which is quite difficult to detect using our current NGS technique. What about your experience, Dr. Bardelli, for utilizing ctDNA detection in the patients with very low tumor volume, especially located in the primary tumor residual area or small lymph node, except liver or bone widespread disease?
Yes, thank you, Dr. Kim. You're raising a very important point, and I think the best way to approach this is to consider these type of analysis similar or parallel to minimal residual disease, when you have to detect post-surgery, for example, whether the curative intent of the surgery has been effective. And you're absolutely right. In there, we face a major challenge, which is technological, okay, mostly. And there are several ways to go about it. One way that we found very effective is because you have surgical tissue, you can design a very high sensitive panel. And in this case, we use multiple single nucleotide variants, somatic, that we use then prospectively to detect whether there is disease left. So one way to go about your question is not to use a standard panel, but to use a patient-specific panel. Of course, this has cost, but we found this to be very effective, and we have implemented this in a large clinical trial that is now minimal residual disease in stage 2 and 3, high risk, post-surgery. Yeah, it's called Pegasus. Thank you for the question.
Thank you very much. Unfortunately, already we used our 10 minutes for the live Q&A. Anyway, it was a very lovely discussion, and we're very happy to have you here. Thank you very much, Dr. Bardelli and Dr. Tsuchihara. Please have a nice lunch and have a good sleep. Thank you very much.
Thank you.
Thank you. A pleasure.
Bye-bye.
Bye-bye.